If we turn our attention away from the developing brain and look at the mature brain, we find that glial cells play a very important role in synaptic transmission. The picture shown here is an electron micrograph of a synapse in the hippocampus, and we see in this picture a presynaptic terminal with multiple synaptic vesicles. This is the active zone right here. This is a postsynaptic process, a dendritic spine. Now, in many synapses in the CNS, we find that these synapses are surrounded by processes of astrocytes. So in fact, this process right here is an astrocytic process. As you can see, it grows right up to the edge of the synaptic cleft. And as we will see in subsequent slides, the astrocytic process plays a very important role in modulating the function of the synapse. Now, these astrocytic processes surrounding synapses are quite common in the CNS. If one looks at the cerebellum, almost all synapses are surrounded by astrocytic processes. If one looks in the hippocampus or the cortex, a good fraction, maybe 50 or 60% of synaptic processes are also surrounded by astrocytes. And for this reason, and for reason that these processes help to modulate synaptic transmission, we like to call the synapse a tripartite synapse. This emphasizes the importance of the third element, the astrocyte, in synaptic function. The intimate association of astrocytes and neurons at the tripartite synapse allows for important communication to occur between neurons and astrocytes at the synapse. And signaling occurs in both directions, from neurons to glial cells, and also from glial cells back to both the pre- and postsynaptic neuronal elements at the synapse. We'll be discussing both types of communication, and we'll first turn our attention to signaling from neurons to glial cells. When neuronal transmission occurs, neurotransmitters are released into the synaptic cleft, and these transmitters not only excite receptors on the postsynaptic neuron, but also receptors on the glial cell, on the astrocyte. And the primary response evoked for stimulation of receptors on astrocytes is an increase in intracellular calcium in the astrocytes. One example of this is shown in this figure. These are recordings of intracellular calcium levels within astrocytes in the retina. The principal astrocyte of the retina is called a Mueller cell, a specialized type of astrocyte. And in order to visualize intracellular calcium, one fills the astrocyte glial cell with a calcium indicator dye that fluoresces brightly when intracellular calcium levels increase. In this case, recordings are made from the retina, and when the retina is stimulated by light, this leads to the release of transmitters from neurons, the stimulation of receptors on the glial cells, and to calcium increases. So in this figure, we see light stimulation down here. This is the retina stimulated by a flickering light, and this evokes calcium transients in several astrocytes that are being recorded from. If we look at the average activity, this is shown in the bottom trace here, where light stimulation is shown to cause a transient and then a sustained increase in calcium in the glial cell. Now in this case, in the retina, it was shown that signaling from neurons to glial cells is caused by the release of ATP from neurons. ATP, one has to remember, is a neurotransmitter or a neuromodulator that is co-released with classical neurotransmitters. So when the retina is stimulated by light, ATP is released from neurons. This stimulates ATP receptors, purinergic receptors in the glial cells, causing calcium increase. A second example of signaling from neurons to glial cells to astrocytes is shown in this figure here. These are recordings from the visual cortex of the ferret in vivo. The same type of technique was used where glial cells were filled with a calcium indicator dye. In this case, both neurons and glial cells were filled with the calcium indicator dye, and neurons were distinguished from glial cells by the addition of a second dye, SR101, which labels just astrocytes. So this is showing an astrocyte. This is showing the calcium indicator dye, Oregon green BAPTA1, where both the astrocyte and a neuron is filled with the dye. The ferret was then stimulated by a visual stimulus to excite the neurons in the visual cortex, and we see with the activation of the neurons, we see an increase in calcium in the neurons and then a delayed increase in calcium in the astrocytes. In this case, the signaling from neurons to astrocytes was mediated by the release of glutamate from the neurons instead of ATP. And indeed, within the brain, glutamate is the primary transmitter that is responsible for stimulating and activating astrocytes. In this particular experiment, the experimenters found that the receptive field properties of the astrocyte match those of the neurons. So, as you know, neurons in the visual cortex can do respond to just a particular type of stimulus, a moving bar oriented in a certain direction, and it was found that the astrocytes responded to the same type of stimulus, indicating that there was very local signaling from the neuron to the astrocyte. So only neurons that were very near the astrocyte actually stimulated the astrocyte and caused a increase in calcium. We have seen two examples of signaling from neurons to astrocytes, but signaling occurs in the opposite direction as well, from astrocytes back to neurons, and this occurs by the release of transmitters from glial cells. This was a rather unexpected and very important discovery that was made maybe 15 years ago or so, that not only do neurons release neurotransmitters, but glial cells do as well. The transmitters released from glial cells are largely the same as those found in neurons. In order to distinguish the two processes, we call the transmitters released from glial cells gliotransmitters, and they include glutamate, which of course is the most important neurotransmitter. This is also prominently released from astrocytes. A second gliotransmitter is ATP. And a third one is D-serine, a rather special amino acid. Most amino acids, as you know, are L-type amino acids, but there are a few D-type. And one of these is D-serine, and this is D-serine is released from astrocytes, and as we'll see in a minute, plays an important role in modulating synaptic transmission. Now, the mechanisms by which gliotransmitters are released from astrocytes is still somewhat controversial, and this question of release mechanisms hasn't been resolved. We know that release can occur by increases in calcium. So just as calcium increases in a presynaptic terminal causes the release of neurotransmitters from neurons, calcium increases in glial cells cause the release of glial transmitters. And one of the mechanisms by which release occurs is by vesicular release via exocytosis, the same mechanism responsible for the release of neurotransmitters. However, Other release mechanisms have also been shown to operate in astrocytes, and these include gliotransmitter release through anion channels and also through hemichannels of these would be gap junctional hemichannels. There are several lines of evidence suggesting that gliotransmitter release from astrocytes can occur via exocytosis. For one, if one looks at electron micrographs of the central nervous system and looks inside astrocytes, you can see vesicles in astrocytes. These are relatively rare compared to the density of vesicles at the presynaptic terminal, but vesicles do occur in astrocytes, and indeed immunohistochemistry shows that these vesicles can contain neurotransmitters including glutamate. The figure shown in this slide illustrates a second type of experiment that has been conducted, which shows that transmitter release can occur via exocytosis. In this case, experiments were done on cultured astrocytes, and the astrocytes were labeled with acridine orange, AO here, and acridine orange is selectively taken up by vesicles. So indeed what we're looking at in this figure here are vesicles labeled with acridine orange. The vesicles were also labeled with a fluorescent V-glut antibody, which labels glutamate-containing vesicles, and one can see that the V-glut vesicles and the acridine orange vesicles are co-localized, and thus they show up as green, demonstrating that the acridine orange is contained in the vesicles in the cultured astrocytes. Now, in these experiments, a special microscopy technique called TIRF was used. TIRF stands for Total Internal Reflectance Microscopy. And with this technique, it's a fluorescence technique that allows one to see fluorescent molecules inside, for instance, vesicles, but only for fluorescent molecules that are within a few tens of nanometers of the surface. So this is experiments were taken with a inverted microscope where the astrocytes were sitting on a glass cover slip, and then using TIRF, vesicles containing a fluorescent molecule, in this case acridine orange, are labeled and visualized, and then as the vesicle approaches the plasma membrane, as it approaches the cover slip, it gets brighter and brighter, and that's what's happening in here. So in this case, the astrocyte was stimulated with a agonist that activated a metabotropic receptor, causing calcium increases in the cell, and this triggered an exocytotic event, which is shown here. The vesicle moves closer to the plasma membrane, and then when it fuses with the plasma membrane and releases the acridine orange, the fluorescence diffuses out and then disappears. And in fact, that's shown here, where the filled circles show the intensity of the vesicle getting brighter as it fuses with the plasma membrane and then releases the fluorescence and then gets dimmer. The open circles are showing a measurement from an annulus, indicating that the fluorescence diffuses outwards as its contents is released. So this type of experiment shows indeed that vesicles in astrocytes can fuse with the plasma membrane, leading to the release of its contents. So indeed, exocytosis and fusion of vesicles can occur in astrocytes. We've seen that astrocytes are capable of releasing gliotransmitters by exocytosis, among other mechanisms. And the first exercise we will be doing in class is the following. Our exercise is to list the ways in which the release of transmitters from glial cells can modulate synaptic transmission and neuronal excitability, and also I'd like you to think about how release of gliotransmitters might affect brain function.